Okay, so welcome to this European webinar series on microglia. I'm Guy Brown from the University of Cambridge, and my co-host today is Jake Dundee. The series consists of monthly talks or 45 minutes by European scientists talking about their research on microglia, followed by a Q&A session. And today's talk is by Claudia Verderio from the University of Milan, Bicocca, and the talk is entitled "The Role of Microglial Extracellular Vesicles in Early Synaptic Dysfunction in Alzheimer's Disease." During the talk, please write any questions you have in the chat function as the speaker talks and direct them to everyone, and then we'll kind of address them, or at least Claudia will address them at the end. But please keep your microphones muted and cameras off to preserve bandwidth. I'll now hand over to my co-host, Jake, who will introduce the speaker.
Thank you very much, Guy. So Claudia undertook her PhD at the University of Brescia in experimental pharmacology or pharmacotherapy. Afterwards, she then moved to Yale University School of Medicine and before becoming a CNR researcher based in Milan. Now she's a CNR Research Director of Biological Sciences, where her research is focused on understanding how activated microglia influences the propagation of synaptic dysfunction and neurodegeneration. Throughout the last 25 years, Claudia has characterized different mechanisms of secretion by both neurons and glia and studied how glia modulate neurotransmission. She's also identified extracellular vesicles as a powerful mediator of intracellular communication in the brain, and specifically the pathogenic role of glia-derived extracellular vesicles in brain inflammation and their potential as a biomarker for inflammatory and degenerative diseases. Claudia's now, like I said, Claudia's now going to give a talk titled "The Role of Microglial Extracellular Vesicles in Early Synaptic Dysfunction in Alzheimer's Disease." So with that, take it away, Claudia.
Okay, so thank you very much for this very kind introduction, and thank you, Guy, for the invitation. I'm really happy and honored to participate in this successful series of webinars on microglia and to share with you our recent finding on the role of extracellular vesicles in amyloid-related synaptic dysfunction in Alzheimer's disease. So, as you all know, microglia and neural communication occurs on multiple levels, ranging from direct physical membrane-to-membrane contact to indirect communication via release of soluble factors. And these factors can be released as single molecules or as multiple factors in association with extracellular vesicles. And so, as introduced by Jakob, the interest in my lab during the past years mostly focused on the role of extracellular vesicles produced by microglia in neuron microglia communication. So what I would like to do today is to briefly summarize what we have learned during the years about the capacity of this vesicle to modulate neurotransmitter release. And I will then describe more in detail the result of two recent studies, one made by Giulia D'Arrigo in the lab, because it provides the rationale for exploring a possible role of extracellular vesicles in spreading of synaptic dysfunction in Alzheimer's disease. And I will then show how another postdoc in the lab, Martina Gabrielli, will address this hypothesis experimentally. So, as you may know, extracellular vesicles are circular membrane fragments released by all cell types that contain a selection of donor cell components such as protein, nucleic acid, and bioactive lipids, and which transfer this molecule from a donor to a recipient cells. And extracellular vesicles are highly heterogeneous in size, but also composition, and can be generated at different subcellular compartment. And two main populations of extracellular vesicles have been described, which are released by healthy cells and which originate at distinct subcellular compartments. And they are called ectosomes or microvesicles or exosomes. So here you see that ectosome, but directly at the cell surface and are quite large in size. They can be up to 500 nanometer or even 1 micron, while small exosomes are generated as intraluminal vesicles inside the endocytic compartment, inside multivesicular bodies, and are released as exosomes upon fusion of multivesicular bodies with the plasma membrane. But once outside the cell, the main function of any type of extracellular vesicle is cell-to-cell communication. So these vesicles can send their messages to the cell they are generated from, to neighboring cells, and through the systemic circulation, they can also reach and signal to distant tissue. And besides being fundamental mediator of cell-to-cell communication, extracellular vesicles during the past years have attracted increasing attention also among neuroscientists for their diagnostic and therapeutic application. And this because during the biogenesis of the vesicles, only a selection of donor cell components are sorted into the vesicles, and their composition changes, reflecting the activation state of donor cells. And this makes extracellular vesicles an important source of information on the activation state of donor cells. And this is particularly important for cells that are not easily accessible, such as brain residence microglia or other cells which are residents in the brain. And in addition, given extracellular vesicles are a natural vehicle for the transfer of molecules between cells, several strategies have been developed during the years to engineer the vesicle surface and to load these vesicles with drugs for selective drug delivery in several therapeutic application. So when we started working on this topic about 15 years ago, extracellular vesicles were much less popular. They were still considered as cellular debris or in vitro artifacts, or alternatively, they were considered as a simple way for the cell to get rid of unwanted material, for example, misfolded protein that could not be properly degraded inside the cells. However, one of the first observations that we made was that production of vesicle, especially from ectosome or microvesicle from the cell surface, dramatically increased upon microglia activation with ATP, as shown in this movie. And this massive release of extracellular vesicle in response to ATP led us to think that ectosome release may be important during brain damage or inflammation, when it's known that the concentration of ATP accumulates extracellularly being released by damaged cells. And during the years, we obtained evidence supporting the existence also in vivo of this vesicle produced by microglia. And for example, this electron micrograph shows a microglia that is infiltrating a myelin lesion in the corpus callosum of a mice. The lesion was induced by injection of lysolecithin in the corpus callosum, and lysolecithin injection induces a myelin lesion that spontaneously recovers over time, providing a robust decision. It was introduced also by Mikhail Simos, the last speaker in this series. And as you see, this microglia that infiltrate the myelin lesion is very dark, is characterized by very dark nucleoplasm and cytoplasm, resembling the so-called dark microglia, a recently described subtypes of microglia that is associated with pathological state. And these dark cells exhibit several blebs on their surface, and it is surrounded by many dark vesicles. And you can better appreciate the budding of this vesicle at higher magnification. In addition, in 2012, in a collaborative study between my lab and the lab of Roberto Furlan, a neurologist at San Raffaele Hospital in Milano, by using flow cytometry and the isolectin IB4 as a marker for microglia and also peripheral macrophages, we could detect myeloid EVs, so produced by microglia and or macrophages, in the cerebrospinal fluid of patients affected by multiple sclerosis. And multiple sclerosis is the prototypical neuroinflammatory disease characterized by microglia activation. And as you see in this graph, the concentration of this vesicle is particularly elevated during relapses and at disease onset compared to healthy subjects. And we then realized that the concentration of myeloid EVs in the cerebrospinal fluid increase in almost any condition, neurological condition, including characterized by microglia activation, including typical neurodegenerative diseases such as Alzheimer's disease or frontotemporal dementia. But although these vesicles in the CSF of myeloid origin are not a disease-specific biomarker, they can also provide, they can anyway provide useful prognostic or diagnostic information. And indeed, last years, Roberto Furlan, who went on measuring by flow cytometry and using isolectin IB4, the concentration of this vesicle in the cerebrospinal fluid of a patient with multiple sclerosis, he found that the simple number of these vesicles in the CSF can predict the disease course of the patients. So being able to distinguish those patients who will develop the disease after a first clinical episode from those who will not convert to the disease. And we also know that the diagnostic and prognostic potential of these vesicles may further increase if we look inside the vesicle and analyze their cargo. And for example, in a recent study carried out by Federica Scaroni in the lab, we measured a set of small non-coding microRNA that target synaptic genes in humans in myeloid EVs, so produced by microglial macrophages, isolated this time not from the CSF, but from the plasma of patients affected by multiple sclerosis. And this analysis was carried out in 2 independent small cohorts of patients. And in both cohorts, patients were distinguished in 2 categories as cognitive preserved and cognitive impaired. And this because a large percentage of patients with multiple sclerosis suffer from symptoms arising from synaptic dysfunction. So they suffer from cognitive impairment. And very briefly, here you see that we found that 2 microRNA, MIR150 and LES7B, are differentially expressed in cognitive impaired patients compared to cognitive preserved in both cohorts. And this suggests that the content of this microRNA can reflect cognitive dysfunction in the patients and may represent a possible biomarker for cognitive dysfunction in these patients. But coming back to basic science that mostly characterize research activities in our lab, here you see the protocol that during the years we have used to isolate a population of medium-large extracellular vesicles produced by microglia in vitro. So very briefly, we truly wash our microglia in primary culture to remove dead cells or damaged cells and to avoid the release into the supernatant intracellular organelle. Then we stimulate microglia with ATP for half an hour to enhance shedding and production of extracellular vesicles, as I showed you before in the movie. Then we collect the supernatant, and by differential centrifugation, after removal of cellular debris, we isolate a pellet of vesicles at 10,000 G for half an hour. And the vesicles present in this pellet are quite large in size, have an average size of about 300 nanometer, and they are highly heterogeneous, however, as indicated by the size distribution of these particles. And in particular, we know that this pellet also contains very small vesicles that are below 100 nanometer in diameter, so that are in the size of exosomes, small exosomes. And these small vesicles can be more easily detected by cryo-electron microscopy. And cryo-electron microscopy indicates that the vesicles are heterogeneous not only in size, but also in their shape and morphology. For example, here you see that most EVs are round in shape and made by a single bilayer. However, there are examples of vesicles that are multilamellar. You see here an example in these slides, and other two examples are shown in this other slide. Moreover, is fraction of extracellular vesicles are characterized by a rough surface that is due to the presence of transmembrane protein or membrane-associated protein that are important for targeting of the vesicle to recipient cells. And there are also a subpopulation of vesicles that are characterized by an irregular shape and even a tubular shape. And the purity of the preparation of the vesicle that we routinely use has been checked also by Western blotting analysis for marker enriched in extracellular vesicle, typical extracellular vesicle marker or negative marker. And for example, here you see that the vesicles are enriched in Alix, flutilin 1 and Nexin 5, while are negative or almost negative for marker of intracellular organelle. So using this preparation of medium-large EVs, mostly released by the cell surface, during the past years, we have analyzed whether this vesicle can influence neurotransmitter release. And we did this analysis using hippocampal neurons in primary cultures or neurons present in cortical brain slices, or also in vivo upon injection of this pellet of vesicles in the visual cortex. So very briefly, we now know that vesicles produced by microglia in primary culture have the capacity to accurately modulate neurotransmitter release, maybe acting on the presynaptic side of the synapse. Specifically, this vesicle can potentiate release of glutamate from presynaptic endings, and this is due to the capacity of the vesicles to enhance sphingolipid cascade in neurons and to induce the generation and synthesis of sphingosine and sphingosine 1-phosphate that increase presynaptic release probability. However, we also know that this same vesicle, the same preparation of vesicle, can decrease the spontaneous release of the inhibitory transmitter GABA at inhibitory neurons. And this is due to the presence and the enrichment also in the vesicles of endogenous cannabinoids, and in particular anandamide, and the activation of CB1 receptor at inhibitory terminals. And given that the capacity of extracellular vesicle to transfer genetic material between cells is probably the most intriguing and fascinating aspect of extracellular vesicle biology, more recently, we also ask whether vesicles produced by microglia can influence neuronal activity by the transfer of their cargo of small non-coding microRNA. And very briefly, what we found is that vesicles produced in vitro by inflammatory microglia, so microglia treated with a cocktail of pro-inflammatory cytokines, through the transfer of a specific microRNA, miR-146A, can induce a postsynaptic remodeling, induce a destabilization of a postsynaptic spine. And miR-146A indeed have many, target many synaptic genes, including neuroligin-1. And neuroligin-1 is a postsynaptic protein that plays a key role for formation and stabilization of the dendritic spine. And through silencing of this protein, downregulation of this protein, the microRNA affect the density of the dendritic spine. And this may suggest that during chronic inflammatory condition, neurons can undergo a pathological loss of synapses. So despite we got some insights during the years on the capacity of microglia-derived EVs to accurately modulate neurotransmitter release and also to induce a postsynaptic remodeling, there are still many open questions to be addressed. For example, we don't know how vesicles produced by microglia move in the pericellular space in vivo to reach target neurons. And we don't know how these vesicles interact with neurons, whether they interact at preferential contact points or whether they can signal in any place at the neuron surface. So what Giulia D'Arrigo did during the past years to try to address this question was to take advantage of optical manipulation combined to time-lapse microscopy and to image the dynamic interaction between a single vesicle produced by microglia and the neuron surface of an hippocampal neurons in primary culture. So here you see that by a highly collimated infrared laser beam that we call optical tweezers, a single vesicle can be trapped and placed on neuron with high spatial and temporal resolution. And we usually keep the vesicle in contact with the neuron surface for 30 seconds, then we switch the laser off. And if a contact is established between the vesicle and the surface of the neuron, we image their dynamic interaction by live imaging in bright field. So using this approach and delivering a very large number of vesicles produced in vitro by astrocytes or microglia, Giulia found that most EVs, the majority of EVs, about 70% of EVs placed on neurons, have the capacity to promptly adhere to any neuronal compartment, the axon, the dendrites, or the cell body. And this suggests that most EVs has in principle the capacity to send a signal to neuron. And quite surprisingly, what Giulia found by looking at the movie that she made during the recording was that a large fraction of vesicles produced by astrocytes, but again also by microglia, after adhesion start moving at the neuron surface, scanning actin protrusion. And this movie shows an example of a vesicle that has been delivered to the growing axon of a developing neuron in primary culture. And as you see, after the contact, the vesicle starts moving, surfing along axonal philopodia during its path towards the growth cone. And given bright field microscopy does not allow to properly distinguish between intra and extracellular vesicle motion, we first of all ask whether the vesicles were moving outside the neurons or were moving inside after internalization. So we reasoned that if the vesicle was still outside the neuron surface, we could retrap the vesicles by the laser trap. So what Giulia did was to select vesicle moving fast on neuronal processes. You see here an example of a vesicle that is moving. And as you see, as soon as she switched on the laser, the vesicle get immediately immobilized. And importantly, the laser trap did not limit the intracellular motion of vesicle actively transported along the processes. So we repeated several times this experiment, and we were able to recapture the vesicles in 10% of cases, meaning that the vesicles were still outside. Sorry. And consistent with an extracellular vesicle motion, we also saw examples of vesicles that were moving from one process to one another, as shown in this movie on the left. And in a few cases, we also saw example of vesicles which could move between processes belonging to distinct neurons. And this suggests that extracellular vesicles may actually use neurites as routes to move among synaptically connected neurons. Okay, so then Giulia tried to also analyze the feature of extracellular vesicle moving at the neuron surface. She mostly did this analysis analyzing vesicle produced by astrocyte, but the features are the same also for vesicle produced by microglia. So she drew the trajectory and analyzed the motion. And what we know is that extracellular vesicles typically display a stop and go motion and a back and forth motion. And interval between period of motion are indicated by the zero value in this plot of the distance run by the vesicles every 100 milliseconds, while back and forth motion is evident in the temporal plot of the distance from the contact points, which shows both positive and negative variations. And with this feature, back and go and stop and go motion, we know that the vesicles mainly move in one direction, that can be either anterograde toward the growth cone or retrograde toward the soma of the neurons. And here you see that the average speed of microglia-derived EVs is about 50 nanometer per second. And this means that a vesicle in 20 minutes can run a length of about 60 micron. Then we ask, how can vesicles move outside neurons? So one hypothesis is that the vesicle binds to a neuronal receptor that is coupled to a moving cytoskeletal network. And by a similar mechanism, viral particles have been recently shown to move at the surface of infected cells to reach the points where these viral particles enter the cell cytoplasm. Alternatively, extracellular vesicles may have an independent capacity to move along a gradient of neuronal receptor, and this would be possible if the vesicle itself contains elements of the cytoskeleton, as well as a source of energy to allow rearrangement of the cytoskeleton, which can elicit in a vesicle motion like in a cell. So very briefly, to distinguish between these two hypotheses, what Julia did was to intoxicate neuronal culture with a metabolic inhibitor, rotenone, or to mildly fix neurons with methanol. And here you see that both treatments produce a strong decrease in the fraction of vesicles moving along neurites, suggesting that the motility of most EVs is passively driven by metabolically active neurons. Then she used different agents, pharmacological drug, targeting the actin or the microtubule cytoskeleton. And here you see that only blebistatin, a cytochalasin D that interfere with actin dynamic, produce a significant decrease in the fraction of vesicle moving at the neuron surface. And this suggests that the receptor of the vesicle is coupling the vesicle to the actin cytoskeleton. And consistent with the existence of a receptor for the vesicles that couple the vesicle to the actin cytoskeleton, we also saw example of vesicles which were induced to move on the neuron surface by the transit of large organelles inside thin processes. So in this movie, the extracellular vesicle is indicated in white, while the dark arrows indicates vesicles that are moving inside the process. And as you see, as soon as the vesicle, the organelle moving inside the vesicle reach the site of contact of the extracellular vesicle on the neuron surface, it generates synchronous motion. And this suggests that the organelle, during its transit inside the process, may drag the cytosolic portion of the receptor, thereby causing synchronous extra and intracellular motion. Okay, but it's also important to point out, and this is another example that shows exactly the same. It's important to point out that we never observe a complete block of extracellular vesicle motion, neither when neurons were fixed with methanol. So this left open the possibility that a fraction of extracellular vesicle can move independently. And as said before, independent motion of extracellular vesicle would be possible if the vesicle itself contains elements of the cytoskeleton and also an energy source. And here you see that by cryo-electron microscopy, we indeed detect actin filament inside a subpopulation of large EVs. And this actin filament that you can see here at higher magnification were frequently present in vesicle characterized by a tubular shape or in a regular shape, but actin-like filament were also visible inside vesicles that are typically characterized by a round shape. We then ask whether the vesicle may contain ATP as a source of energy to support actin dynamic. So we measure the content of ATP in the vesicle by two bioassays. In the first bioassay, the ATP sensor cells are loaded with the calcium-sensitive dye FURA2 and display calcium transient in response to ATP. And here you see that addition of microglia-derived EVs to the sensor cell produce a clear calcium transient, which was completely blocked in the presence of the ATP-degrading enzyme apirase, indicating that ATP was the agent causing this calcium transient, and indicating that the vesicle contain ATP and also leak ATP into the extracellular space to activate the receptor. And in the second bioassay, we use B16F10 cells that are stably transfected with a firefly luciferase on their membrane. And these firefly luciferase emit light in response to ATP. And again, addition of large EVs produced by microglia to the sensor cells produce a significant increase in light emission, so confirming the presence in the vesicle of ATP and also the leakage from the vesicle lumen into the extracellular space. So having found that a subpopulation of large EVs contain actin filaments and that this vesicle likely contain ATP inside their lumen, we next ask whether dynamic rearrangement of the actin cytoskeleton could be important for the motion of a fractional extracellular vesicles. So this time we treated the vesicle with cytokalasin D. This drug induced a depolymerization of filamentous actin in globular actin, as indicated by the Western blotting analysis. And this analysis produced a significant decrease by 30% of the fraction of vesicle moving at the neuron surface. And so this indicates that about 1/3 of the vesicles moving at the neuron surface may have an independent capacity to move. So to sum up this first part of the presentation, our data suggest a complex and unexpected trafficking of glial EVs produced by astrocytes and microglia at the surface of neurites, and provide initial evidence for the existence of a dual mechanism, a passive and an active mechanism of extracellular transport. And the existence of such complex trafficking of the vesicles at the surface of neuron in primary culture suggests that also in vivo extracellular vesicle may move not randomly, but may use axonal projection to move among synaptically connected neurons. And given extensive literature indicates that under neurodegenerative condition, extracellular vesicles become vehicle of misfolded protein, A-beta and tau protein in Alzheimer's disease, alpha-synuclein in Parkinson's disease, ATDP43 in ALS, the possibility opens that motion of vesicle carrying misfolded protein and long axonal projection may have a role in the spreading of neurodegenerative lesion that propagates in the brain of patients affected by neurodegenerative disease over time. So we decided to address this hypothesis, and what Martina Gabrielli did was to explore whether vesicle produced by microglia and carrying amyloid species could have a role in, could cause and propagate alteration in long-term potentiation, a form of synaptic plasticity that is thought to underlie learning and memory. So this slide is just to remind you that Alzheimer's disease is the most common form of human dementia in the elderly, characterized by loss of memory, reasoning, but also behavioral abnormalities. As you all know, the disease, all marks of the disease are extracellular amyloid plaque, intraneuronal aggregate of hyperphosphorated tau, and neuronal synaptic loss. However, at early disease stages, the main alteration is a functional alteration and is in alteration in synaptic transmission. And interestingly, synaptic dysfunction first occurs in the region where later on in life aggregates of tau and also of A-beta will appear and will propagate through the brain of patients following the same anatomical pattern of connection. However, how synaptic dysfunction starts and propagates is still unclear. And finally, the last hallmark of Alzheimer's disease is microgliosis. The level of microglia activation increases linearly throughout disease progression and positive correlates with cognitive decline in patients. And importantly, in recent studies, have shown that microglia activation may even precede plaque formation in a transgenic APP rat models, unveiling an unexpected role of microglia activation also in early synaptic symptoms and synaptic dysfunction in the disease. And so far, two main mechanisms have been described by which reactive activated microglia may contribute to synaptic loss in Alzheimer's disease. That is excessive synaptic pruning, excessive removal by phagocytosis or partial phagocytosis of synapses, or the release from reactive microglia of extracellular vesicles that contain neurotoxic tau or A-beta species. And the latter observation was made among other groups also by our labs. And in 2014, in a collaborative studies between my lab and the lab of Michela Matteoli at the Neuroscience Institute of Milano, we found that microglia in primary culture efficiently internalize the human amyloidogenic peptide A-beta 142, but is also able to resecrete this A-beta form in association with extracellular vesicles. And in this confocal picture, A-beta is staining blue, while in green you see the staining for the isolectin IB4 that stain also the surface of the vesicles. And the presence of amyloidogenic cargo, A-beta and A-beta 140 and A-beta 142 has been confirmed also by cell death of mass spectrometry and Western blotting analysis, and more recently also by a specific ELISA. So the very early activation of microglia, which even precedes plaque formation in transgenic model of the disease, the presence of A-beta form in the vesicle and the capacity of this vesicle to move along axonal projection prompt us to investigate whether this vesicle may have a role in early synaptic dysfunction and in their propagation within throughout the brain. And here you see the protocol that Martina Gabrielli used to isolate a population of vesicle-carrying A-beta species that we call A-beta-Vs. So before isolating by differential centrifugation on the pellet of the vesicles, as previously described, she incubated microglia in primary culture with the human A-beta peptide, A-beta 142, or alternatively, she incubated microglia in primary culture with soluble A-beta species naturally secreted from CHO cells transfected with a mutant form of APP. And first of all, Martina explored the dynamic of the motion of control EVs and A-beta Vs along the axon of the hippocampal neuron in primary culture. So using optical manipulation and time-lapse microscopy, she placed single vesicles along the axon of neurons, and axons were selected based on their thin morphology and the absence of dendritic spine. And here you see that she found that vesicle carrying A-beta were more prone to motility. So twice the number of the vesicle move along the axon. The average speed of the vesicle was significantly higher. And importantly, vesicle carrying A-beta species mostly move in an anterograde direction, so toward the growth cone. And this would be consistent with a transsynaptic spreading of a pathological signal. And Martina also found that it is possible to limit the motion of this vesicle along the axon by pretreatment of the vesicle with annexin 5. And annexin 5 is a ligand of phosphatidylserine that is externalized on the surface of quite large vesicles produced by microglia. And this treatment does not prevent the interaction of the vesicle with neurons. It rather promotes the adhesion of the vesicle to neurons, but it reduces by 50% the fraction of moving vesicles. Then we ask what might happen when a vesicle, by moving along the axon, reach synapses on a postsynaptic dendrites. So we directly place control EVs or A-beta Vs on secondary dendrites in hippocampal neurons, and Martina measured the density and the shape of dendritic spine at the contact site and far away the contact sites. And what she found is that both vesicles containing A beta or control vesicles were able to induce an increase in the density of dendritic spine. However, while control vesicle produce an increase in the density of mature dendritic spine, so characterized by a mushroom shape, vesicle carrying A beta produce an increase in immature dendritic spine, so cause a shift in the spine shape towards an immature morphology. And given that the size of the dendritic spine is a structural correlate with synaptic strength and can reflect synaptic plasticity phenomena such as LTP and LTD, we then ask whether vesicle carrying A beta may actually interfere with long-term potentiation. So first of all, Martina incubated hippocampal neuron with control EVs or A beta EVs. Then she washed the culture, and by whole-cell patch clamp recording, she measured miniature excitatory postsynaptic currents before and after induction of synaptic plasticity. As synaptic plasticity was induced by a chemical protocol that relied on a brief application of glycine in magnesium-free medium. And the yellow line indicates that neurons pretreated with A beta EVs completely lost their capacity of undergoing a long-lasting increase in MAPS frequency compared to vehicle-treated neurons or neurons treated with control EVs. And this indicates that the amyloid cargo of the vesicles interfere with synaptic plasticity in vivo. So encouraged by this finding, we then ask whether A beta EVs may interfere with long-term potentiation in the entorhinal hippocampal circuits, a key site for memory formation that is particularly affected in Alzheimer's disease. And here you see that these circuits start in the entorhinal cortex and through connection called the perforant path project to the dental gyrus. So what Martina did was to inject control EVs, A beta EVs, or synthetic oligomeric peptide as control in the entorhinal cortex, and using cortical hippocampal slices, she induced and measured LTP at the injection site and in its main target region, the dental gyrus. And these measurements were done 1 hour after injection of the vesicle in the entorhinal cortex and 24 hours later. And here you see the result. So 1 hour after injection of the vesicles in the entorhinal cortex, LTP was completely impaired, as indicated by the orange line. Sorry, the yellow line, 1 hour. At this time point, LTP was reliable elicited in the contralateral side that was injected with control vesicles. And at this time point, LTP was also reliable elicited at the synapse between the perforant path and the dental gyrus, as again indicated in yellow. However, 24 hours later, as indicated in orange, LTP became impaired also at the synapse between the perforant path and the dental gyrus. And this indicates that a single injection of A beta EVs in the entorhinal cortex is able to cause an LTP impairment that propagates along the circuits to the dental gyrus. And finally, we ask whether we could interfere with the spreading of synaptic dysfunction along these circuits by limiting the motion of large EVs along axonal projection. So what Martina did was to inject A beta EVs pre-treated with annexin 5. I've explained before that this treatment limits by 50% the motion of the vesicle along the axon of hippocampal neuron in primary culture. So as expected, Martina found that vesicle pretreated with annexin 5 produced a block of LTP at the site of injection, 1 hour after injection. However, as you see on the right in red, LTP was still elicited 24 hours later in the dental gyrus, implicating the motion of this vesicle along axonal projection in the spreading of synaptic dysfunction along the circuits. So this is my last slide. So our data provides strong evidence of the involvement of quite large vesicles produced by microglia in primary culture in the onset and propagation of synaptic dysfunction. we have shown that vesicle produced by microglia preloaded with A-beta, so storing A-beta species, cause impairment in LTP, both in vitro and in vivo, along the entorhinal circuits. And despite we do not provide direct evidence of extracellular vesicle motion in vivo in the mouse brain, this is mostly due to the difficulties in imaging small vesicles in a complex structure such as the mouse brain. Our data strongly suggest that this motion of large vesicles can have a role in the propagation and spreading of synaptic dysfunction along the entorhinal circuits. Therefore, we really think that a better understanding of how these vesicles move at the surface of neurons along axonal projection open up novel strategy to delay the onset and limit the propagation of synaptic dysfunction in Alzheimer's disease. And here you see the people in the lab who did the work. I've already mentioned Giulia D'Arrigo and Martina Gabrielli, Federica Scaroni, also for the analysis of microRNA in vesicles from patients with multiple sclerosis. The patients were provided by two groups, one in Amsterdam and one in Milano. And here you see the very crucial collaborator that helped us with two recent studies, Giuseppe Legname and Dan Kojok in Trieste. Francesco Di Virgilio in Ferrara, which helped us in detecting ATP in the vesicles, and a great contribution also in the design of the experiment and in performing the experiment by Nicola Origlia of our institute in Pisa and Ottavio Arancio at the Columbia University in New York. And here you see also a list of our collaborators, which greatly contributed to define the role of extracellular vesicles in glia-neuron communication and also in the communication with oligodendrocytes during the years, and our previous lab member. So I think I'm at the end, and thank you for your attention. I'm willing to take your question.